certainly a pleasure to be here. And since it was mentioned that I was recruited by Barack Obama, I have to give a brief disclaimer. I actually, believe it or not, still work at the White House. And what this means is that I'm required to give you a little warning, which is that this is a personal talk. This has not been approved by White House Communications. I am not representing the United States government. And this talk may not reflect the views of the president or the administration. What I can tell you is that it used to. The next disclaimer. So as it was mentioned, I have a PhD in computer science. In fact, I have about a grade school education in biology. And I'm about to give you a lecture on medicine. So I spent most of my professional career actually working in computer science, so things like cybersecurity, supercomputing, and fundamental algorithm and data structure design. So with that disclaimer aside, I want to define precision medicine because this fits very nicely with this theme of delivering the right care to the right patient at the right time. This is very much the essence of precision medicine. So I think Barack Obama defined it very well when he launched the Precision Medicine Initiative. So when he launched it, he said it's really about data-driven medicine. If you're using data to optimize the health care of individual patients, you're doing precision medicine. Now, at the time, there was a new piece of data starting to being used in the clinic, which we're still learning how to use in the clinic, which is the genome. So these days, precision medicine and genomic medicine are often synonymous, but they're not necessarily the same exact thing. So I think precision medicine is much bigger than genomic medicine because it includes all forms of data that you can use in optimizing the health care of your patients. And again, I think the third key point to define precision medicine is that this is all about delivering the right drug to the right patient at the right time. That's the essence of precision medicine. So in a nutshell, we take the patient, we map them into their data, which again often includes their genome. We find some sort of mutation driving their disease. And then we use this information to find the perfect medication for them, or the best possible medication we can give them right now. That's what we're trying to do in precision medicine. So there's 3 points I want to get across in this talk today. The first thing I want to do is tell you a story. It's a story of what happens when you're told that your child is the first case ever discovered of an ultra-rare genetic disorder, as I was 5 years ago. The next thing I want to do is use the story to illustrate an algorithm for doing precision medicine, for performing these diagnostics and finding these therapeutics for patients today. And the third thing I want to do is give you a vision for what clinical precision medicine is going to look like. And I'll do this by describing some of what's happening at my very new institute at the University of Alabama at Birmingham. So to start, I'm going to tell you a little bit about myself and really about my son Bertrand. So this is Bertrand. He's almost 10 years old today. This is him, I think, back at about a year and a half old. And the key thing I want to get across here is that even though I'm telling you a story about Bertrand, this is by no means unique to Bertrand. This is a story being told many, many times over for basically every rare disease patient in the world. And it's a story that often begins on a place called Undiagnosed Island. This is an island with a population of one patient or one family. And when you're trapped here, it's a pretty daunting place to be. When we were there on Undiagnosed Island, we didn't know why it was the case that Bertrand was having severe developmental delay. We didn't know why he had seizures. We didn't know why he had a strange movement disorder. We didn't know why, for some reason, he could not cry tears. Now, he could cry and go through all the emotions that you associate with crying, but no tears would ever come from his eyes, which in fact was leading to very dry eyes and some medical complications from that. So we were trapped there for 4 years. For 48 months, we were stuck on Undiagnosed Island. And that was until we started collaborating with a team of scientists at Duke University. And I'd learned enough about what was going on with Bertrand at this point to know that, I was learning a lot of genetics. We felt it was a genetic disorder. I thought, okay, we probably need to look into his genome to find the answer to this disease. At the time, this is 8 years ago, you really couldn't get clinical genomic sequencing. It just wasn't something you could do very easily. But then working with this team at Duke University, this new technique, exome sequencing, came along. We said, we think we can use this to very economically find the cause of this disease. So we partnered with them, and we did what was called trio sequencing. In trio sequencing, we sequenced myself, we sequenced his mom, we sequenced him, and we looked for mutations that could potentially be causing his disease. And what we found was that he did in fact have 2 mutations that looked very suspicious. It looked like he had 2 mutations completely knocking out the function of a gene called NGLY1 or NGLY1. And then with this information came some very interesting news. They said, it appears to be the case that Bertrand is the first patient we've ever seen with this disease. If this is really the cause, we think this is the first case ever. So he's the only one we know of. He's in many ways an N of 1. As a result, there's not a whole lot we can do to tell you about what's going on with this disease. We can't even give you a prognosis in the absence of other patients. So the real question is, and oftentimes in precision medicine, is how do you know that this mutation, because when you do this genomic sequencing, you find lots of mutations in patients. The question is, which one is really the culprit? And how do we know this? How are we confident that this is the one? Well, there's a process here called variant interpretation, where you look at all the mutations, you say, well, how could these drive the disease? To do this, you have to take the aggregate picture of the patient, usually collected from the medical records, and map this onto the underlying purported genotype or mutation driving the disease. And to do this, in every case, you have to understand what that gene is doing. Now, this gene, N-gly1, is responsible for encoding an enzyme called N-glycanase. And N-glycanase is responsible for deglycosylating misfolded glycoproteins after they have been retrotranslocated from the endoplasmic reticulum into the cytoplasm. So obviously, what's happening here, I'll pause there and just say that, as a computer scientist and as a parent, there are sentences I did not expect to have to say. That was certainly one of them right there. So I was not by any means an expert in glycobiology, but what's really going on here is we found out Bertrand is missing a recycling enzyme that deglycosylates and decommissions misfolded glycoproteins. So if you take this enzyme away, you would expect to see a certain kind of trash accumulating in the cells. And the trash in this case is misfolded glycoproteins. So the question was, do we see this happening in patients? Or do we see this happening in Bertrand? And we partnered with a glycobiologist. We looked at his medical records, and we found evidence that, in fact, yes, there was this kind of process happening in his liver. We said, at this point, this is almost certainly the cause of this disease. And in the absence of finding any more patients, we really couldn't be totally certain that this is what it was, but this is as close as we could get at the time. But then, of course, the words not actionable pop up. They say, there is nothing that you can do in this situation. There is no medication. There's no prognosis. No drug company will ever care about this. So there's really nothing more than you have than the comfort of having a name for this disease at this point. But the thing is, I'm not sure that was true then. I don't think it was ever really true. It's certainly not true now. I think just going from the dozens of potential diseases this could have been down to one overnight, that in itself was a major actionable event. Because the truth of the matter is, if you give a parent a target to shoot at, a parent will go collect arrows. That's what we do on behalf of our children. And it underscores a key theme that I think is popping up again and again in this notion of precision medicine, which is that science is what becomes action in the event of the inactionable. There's no medication you can take, and what you do is science. And so science itself becomes a kind of medicine in the absence of any medication you can take. So, in fact, within just weeks of the discovery of this disease and getting this diagnosis, we were actually taking the first credible attack on the disorder itself. So there's I won't get into the details of this in terms of what's happening molecularly, but there's a class of compounds out there that's capable of attacking certain kinds of mutations. In fact, the kind of mutation he happened to receive from me. It's called a premature stop, where there's basically a little stop sign in the middle of this gene, and it's stopping production prematurely. Well, there's a class of compounds called read-through compounds that can, in theory at least, interfere with this stop sign and say, keep going, go ahead and make the full protein. So we tried these on his skin cells at that lab in California, where we were working with Dr. Hudson Freeze, and just to see if any of them would actually work. Now, none of them did. Some people say, was that discouraging? Well, I was like, no, not really. Because to me, it was still amazing to think that we could go from having no clue what this disease was to trying something at all in just a matter of weeks. And so that was encouraging. It said, let's keep going. Let's keep doing this. So it said, let's do some science. Let's see how far we can go. And of course, to do science, you need to stand on the shoulders of giants. And in this case, when we searched for giants, we didn't find very many. So if you looked at PubMed 5 years ago when this disease was being discovered, you'd find 5 publications on this gene. So we as a species didn't understand a whole lot about what this gene was actually doing. And what that meant was we had to go find more giants. And to do this, we started partnering with a glycobiologist in California, Dr. Hudson Freeze, who sort of understood what this gene was doing and could paint a picture for us of where, just how far we had come ultimately on this diagnostic odyssey. But more importantly, from this vantage point, what we just learned, how far we would have to go if what we wanted to do was ultimately reach understanding, treatment, and cure. And we realized very quickly that as challenging as it had been over the past 4 years to reach just a diagnosis, it would be significantly more challenging still to find a treatment for this disease. But as a parent, you don't really think about how far you have to go. You just sort of say, well, we'll just take one step after another. And I've been taking steps now for 5.5 years. And what follows is what happens if you apply that philosophy over and over and over again. But we realized that there's no way you can do this alone. This is one family versus one disease that just doesn't work. So we have to find others. And I realized that there were probably others out there by looking in genomic databases that were available at the time to predict roughly how many patients there should be in the world. I could look at how many other damaging mutations there were in this gene out there in the population and get a crude estimate how many other patients there should be out there somewhere. And I realized that this is a very rare disease. It's a 1 in millions kind of disease. However, there should be about 500 patients out there somewhere that have it. They just don't know what they have. So to find them, I wrote a blog post. And this blog post had to do 2 things. It had to go viral, and it had to rank very highly in Google search results so that if other patients or parents were out there searching for it, they would find it. That was the idea. So this is the blog post I wrote. There's a couple things to point out here. The picture of Liam Neeson, that's to make it go viral. And I often think that I could put a cat picture there as well, and it probably would have also gone viral, because the internet is kind of sadly predictable in these ways. There's 5,000 words beneath this that are designed to essentially instruct Google on how to rank this page, so that if other patients or parents are out there searching for things like lack of tears, this page will pop up, or is much more likely to pop up than others. Because again, that's the idea, help other patients find us. So in fact, it did go viral. Very quickly, it was seen by millions of people, and it started ranking very highly in Google search results for all the right terms. So remarkably enough, other patients did start to find us. They started finding us very quickly. So within just a couple of weeks, we actually found 2 more patients in Turkey. And in fact, the next 2 patients after that were found a few months actually here in Israel. So patients started popping up all over the world almost overnight. And what this did for us to go from no disease to 1 patient to a community in just a few short months was really astonishing. What it did was it really washed away this dark night of uncertainty that you live with on an undiagnosed island and replaced it with the sunlight of science and community. And it gave us a way forward. So that community today, I can tell you, is extraordinarily united in finding, understanding treatment and cure for this disease. And in fact, we now have 59 patients around the world diagnosed with this disease. So over 10% of the patient population is working together and connected to fight this disease. And now we can ask questions like, well, what can you do with an N of 59 that you could never have done with an N of 1? And it turns out there's a lot you can do. So we got very lucky that around the same time this disease was being discovered, the NIH was very interested in studying disorders of glycobiology. And we argued very coherently that this was, in fact, a brand new disorder of glycobiology and glycosylation, and that they should be very interested in studying this. And as soon as they said yes, we took essentially every living patient and pushed them into this research protocol. And what you do in this research protocol is every patient spends 1 week every year indefinitely being deeply studied by scientists at the NIH. So I'm very grateful to Bill Gall for allowing us into this protocol. And to show you what it's like, I have a picture here for every post-prod procedure or appointment that occurs during the course of this week-long admission at NIH. So this is the NIH Clinical Center. It is a very intense week every single time you do it. We've been doing this now for 3 years. And the value in doing this again and again, and having this kind of buy-in from the patient community, is that you get tremendous amounts of data. So we already, within a year and a half, found a urine biomarker for this disease. We found a unique sugar in the urine of these patients that definitively diagnoses the disorder. So we don't need genomics anymore to diagnose this disease. We can do it with urine. And we are finding patients now with urine because you can get this test from the Mayo Clinic or from Emory. This also is serving as a surrogate endpoint for something like an FDA clinical trial. So we can say, here's something we can measure to measure the effectiveness of therapies for this disease. So the question is, how do you go from all the understanding we started building up by doing the science into actually treating a disease? This is the key part and most challenging part of precision medicine, and really I think of the broader theme of an event like this. And I think the key answer is you want to screen drugs. You want to start taking the existing drugs that are out there and finding new ways to use them for all of these diseases or for individual patients. And but how do you do this? Well, here's the high-level approach. You take the patient, you find the genetic insult driving their disease, and you turn this information into some sort of model. Oftentimes it's a cellular model. That model might even come directly from the patients themselves. And what you find is a way of measuring whether or not this model is doing well or poorly. So you find a measure of sickness for the model, and then you grow it up into as many cells or as many model organisms as you can possibly grow, and you start combining it with as many drugs as you can combine it with, usually the entire FDA-approved library in our case. And then you check to see if any drug out there actually makes a difference for this particular patient in this model. If you find a drug that seems to reverse it at the model level, then you have some hope that this drug might work at the patient level. And so you now have a way of giving a drug to the patient. So that's sort of the essence of a lot of precision medicine right now. And a big part of this, again, is creating models, everything from cellular models, but also including things like animal models. And so the other thing that happened around this time this disease was discovered was that CRISPR was being co-discovered. And CRISPR has made it dramatically cheaper to create model organisms overnight, not just in time, but also in cost. So for Engli 1, we have mice, we have flies, we have zebrafish, we have worms, and we even have yeast. Now, I don't know if our yeast have the same wonderful nutty flavor as those depicted here, but we certainly have yeast available for Engli 1 today. And all of these are giving us insights into the nature of this disease that would not be possible without them. Now, the other thing you can do is you can say, well, if you know the exact protein that's missing in these patients, in this case it's an enzyme, can you just put the protein back in as a drug? Can you just give it as if it were a medicine? Well, in theory, you can. So you can synthesize Engli 1. You can think about putting it back in cells. In fact, you don't even have to go through the process of synthesizing. You can just buy it on the internet. So if you Google Engli 1, you can buy human Engli 1 online, and it only costs about $402 for 10 micrograms. Now, the only problem with that is if you work this out to therapeutic quantities, it's about $2 million a week, which is extraordinarily expensive. And I actually asked my old boss, is this covered under the Affordable Care Act? And he went, no, Matt, it's not covered. Even then, it's not exactly perfect. So another problem here is that most proteins don't naturally act as drugs. They act as food in many cases. And so if you just inject this thing or if you eat it, then it'll end up in the lysosome or digested. So you have to modify this thing through a process like pegylation and then TAT tagging to make sure it actually lands in the right part of the cell. So this is hard. And it turns out there's easier things you can consider in many cases before doing that. One of the easy things you can consider is a metabolic diet. If you know the exact genetic cause of the disease, if you know that root cause, maybe you can modify diet in some way to address this enzymatic deficiency. So the key idea here is if you have an enzyme that turns A's into B's, and suddenly it's gone, then maybe what's driving the disease is you have too much A, or you have too little B. So maybe you can modify the diet of the patient so that what you're doing instead is you're giving B as a supplement, or you're restricting A or the precursors to A so that they don't get this toxic accumulation. And there's other diseases out there where this works very well. So for example, for PKU, if you restrict phenylalanine, it's effectively a cure for the disease. If you don't, it's a devastating disorder. Same is true for MPI-CDG. If you give mannose as a supplement, it's a very treatable disease. If you don't, it's a devastating disorder. And the question is, what does this mean for Engli 1? It's also an enzyme. It has A's and B's. Can we figure out what to restrict and what to give? And through some advances in basic science around this disorder that began right around the discovery of this disorder, when we started funding a postdoc to work on this, we had an idea of what those A's and B's were. And eventually, I put this picture together that said that in this particular case, there's a structure to the way this waste is accumulating in the cells. And in fact, it appears to be sequestering, or probably sequestering, one metabolite in particular. That metabolite is represented by here by this little blue square. It happens to be called N-acetylglucosamine. And if you Google N-acetylglucosamine, what you'll find is that you can buy it on the internet. So that's what I did. So I said, I think my son is deficient in this little blue square. I can buy it on the internet. And then this little bottle arrives in the mail. And just to make sure it was safe, I took the whole thing myself. Now, I'm still here, and as a result, I figured that constituted FDA Phase 1 safety testing for my household. And this is where I have to remind you that I'm not actually the FDA. I don't represent the FDA. I'm speaking on behalf of myself. So, then there was sort of a broader ethical dilemma, which is, well, what do you do? I mean, so okay, I took it, I survived, but can I give this to Bertrand? And he kind of answered that question for us. He was prone to recurrent pneumonias, and he ended up with a very serious pneumonia. At one point, I thought he might actually not survive his hospital stay. And I thought, if he had not survived this, and this little bottle was sitting on my shelf, and I had never figured out if it worked for him, there's no way I could forgive myself. So I said, as soon as he's home, as soon as he's better, I'm going to put him on this compound. And so that's what we did. And about 3 days after he went on this compound, for the first time in his life, and in fact the history of this disease, he cried tears. So I walked in this morning, I heard him crying, because he was hungry for breakfast, and there were tears coming down his cheeks. So I had no idea what to expect when this happened. I didn't know what to look for, but this was a very clear sign to me that something was different. Something was happening with this compound. And in fact, over the course of the next 3 months, his nighttime seizures, which were very different from his daytime seizures, actually went away completely, and they have never come back. So this is a major quality of life improvement for him. And the thing I always tell people is, when you're a parent in a situation like this, you sometimes do strange things. So when I actually saw him crying those tears, I think I did what most of us would have done in that situation, which was I collected his tears, I packed them on dry ice, and I shipped them to a lab in California for analysis. I don't do that with his tears anymore, but the thing is, you can't leave any science on the table. And so that they may have been small tears for Bertrand, but they turned into an ocean of science for the disorder as a whole. And the other thing I find funny is when I give this talk to many audiences, they say, well, that's wonderful that you found something that works for him. When I give this talk to biologists or to people in medicine, they often say, well, it's great it works for humans, Matt, but what we want to know is, does this work in flies? And so we went ahead and gave NGLI1 deficiency to flies. And it turns out if you do that, they have about an 18% survival rate. It's a pretty lethal mutation in flies. But if you raise them on this compound, their survival rate rises to about 80 to 90%. So we said, okay, there is something here that is really making a difference for these patients. And these flies have now contributed greatly to the science of this disease and our understanding of it. At this point, this is maybe 3 or 4 years ago, I was really started shifting more and more from computer science into biology and medicine. I mean, obviously my attention was captivated here. I was motivated here to do this. And so I said, well, we've come this far. How far can we go? Can we go all the way? And I actually picked up a grant to do novel drug development for NGLI1 deficiency and partnered with Kubi Balaguranathan, who was one of my colleagues at the University of Utah at the time. And to do this, what we did is, okay, let's make a new model for the development of this disease. So we started working with planarian worms. And the cool thing about planarian worms is that if you cut a planarian worm in half, what you get is 2 planaria. They're fully regenerative. So they're an excellent model of development. Now the great thing is, if you take a planarian worm and you give it NGLI1 deficiency, do you know what you get? What you get is a worm that's been cut in half. So they've completely lost their regenerative capacity when you give them NGLI1 deficiency, which meant that they're a very good model for development in this disease. So we started knocking out other genes in conjunction with NGLI1 in the hopes that one of them would actually reverse this disease. And in fact, doing this again and again, we eventually found one, a gene called NGase, which when knocked out in conjunction with NGLI1, in other words, by taking out 2 genes, we actually made the disease better. This just happens sometimes because biology is weird. And we were looking for that gene when knocked out in conjunction, which would make it better. And what that meant is we had a drug target. We had an inhibitor that we wanted to create to make this disease better. So to find an inhibitor for this gene NGase, I went from biology and medicine back towards computer science. And we created a homology model of the crystal structure of NGase, and then did computational simulations to find out if anything could serve as an inhibitor to this particular enzyme. And we screened about 200,000 molecules, checking to see if any of them were, again, sort of inverse in shape and charge, the catalytic domain on this protein. And what we found was about 70 compounds appeared to interact with it, which meant they could be inhibitors, and 14 were already FDA approved. So this immediately focused our attention on these 14. We took all 14 to the bench, and we used mass spectrometry to check to see if any of them really were NGase inhibitors and really could be potential therapies for this disease. And one of them actually was. And that one drug, which we found about a year ago, is Prevacid. So Prevacid is available in the US over the counter as an acid reflux medication. Bertrand doesn't have acid reflux, but it doesn't matter. This molecule happens to have the right shape to hit the right target to make his condition better. So the side effect of this drug is also a treatment for NGLI1 deficiency. That's the short version here. And so we wrote up a paper on how we found this, if you want to know more about it. But it also plunged us into a process called medicinal chemistry. Because Prevacid, while it is an inhibitor of this target, it's not as potent as it could possibly be. And so we're searching for better, stronger, more potent, less toxic inhibitors than just Prevacid, but based on Prevacid. So we've actually managed to attract the attention of a drug company to do exactly this. So Retrophin, which is a pharmaceutical company in the US, said, you know what, we think you have de-risked the science enough that we will take this forward now and we will help find an inhibitor for NGase based on what you've done with Prevacid. So we're taking Prevacid, we're making modifications to it, and we're generating lots of different molecules based on it that are very similar, which hopefully have the same effect. And what we found so far is one molecule that already is 60 times as potent as Prevacid. The problem is this is a new drug, and it has to get reapproved through clinical trials just like any other drug would. But the point is we've now got a great lead and great path forward for developing ever better drugs for this disease. And this means we have to go through clinical trials. And clinical trials are frankly just hard. So, as hard as it was to get a diagnosis and as difficult it was to reach a treatment after that, more difficult still is to get regulatory approval. And the thing about this third mountain is you don't even see it coming until you get there, and then suddenly there it is. So, we've been lucky in finding ways to try to circumvent this mountain, but ultimately it requires the work of everyone to reach out to their government and say, the science is ahead of the medicine when it comes to precision medicine. We need better laws and better regulations around this. So I've gone to both the House and the Senate of the United States to speak there on behalf of something called 21st Century Cures, which passed in the waning days of the Obama administration, which started to lay some of the foundation for the regulatory structure necessary to enable precision medicine. I wouldn't say we have it exactly right. We have more to do, and I'm optimistic going forward that we'll get the regulations right eventually. And now people always ask, since we sort of zoom forward today, how is Bertrand? And the answer is he's happy. He's a very happy kid these days. He has 2 wonderful younger siblings. And he used to have hundreds of seizures per day, literally hundreds, and now he has none, not a single clinical seizure at all. We were concerned at one point he would go blind from corneal erosion. But now, with the tears he can produce, there's no risk at all of that. And what's sad is that many of the older patients we found did in fact go blind from corneal erosion because there wasn't any treatment available at the time for them. And no matter how aggressively you keep the eyes lubricated, you just can't keep up with this process. But Bertrand's going to be fine. Now, he still has very severe developmental delay. You can't just magically undo that. But what I can see from Prevacid, we have him on the highest safe dose we can get him on, is that he's doing things now that I have not seen him do in 10 years. So I believe that whatever development is left for him, we can unlock that, and we have a strategy for doing that. But again, the bottom line is he's happy, and I think for any parent, that's the ultimate patient-reported outcome, is happiness. So I feel like it's mission accomplished. So to close, I just want to sort of share the vision for how we go from an N of 1 to many N's of 1 over time. And I think to do this, what you really need is an algorithm or a process for scaling this way of going from patient into the right pill for them. And so I won't go into detail on this particular algorithm, but what I will show you is that I have thought through it. There's a process for how you go from a patient to a pill, or a patient to their information, and from their information ultimately into a medication for them. And of course, it always, and usually it branches on things like, what's the genotype of the patient? Is it a gain of function? Is it a loss of function? Is it a change of function? And ultimately, this leads you either to experiments to run or to a list of medications that might work for them. And ultimately into clinical trials at the end of all that. So the question is, can we really do this? Was Bertrand a special case, or is this a process that we can scale up? And I will tell you the answer is yes, and I can say yes because I got some extraordinary opportunities. So around 3 years ago, the Harvard Med School asked me to join as a visiting professor, and I remain on the faculty there to this day. And they said, can you help us scale up your vision for precision medicine in the clinic here? Can you try this out on more patients? So I started doing that. And then, about 2 years ago, this guy asked me to come to his house and said, I read an article about you, and I'd like you to help do for the entire country what you have done for your son. And I said, okay, I will absolutely do that. Whatever you want, anything you ask, tell me what to do. And so I started working on what was soon to be the Precision Medicine Initiative, and after that launch, I started working on something else within the Precision Medicine Initiative, which I announced about a year later, which is this pilot project to see if we could really do this again and again. Because the Precision Medicine Initiative is really about laying the scientific foundation for precision medicine. What we wanted to know was, could we do it in the clinic today? So what we did is we created something called the Patient Empowered Precision Medicine Alliance out of the White House, roping together several clinical organizations that could actually execute this vision I had. And we went after 5 diseases in 12 months. And we picked 4 ion channelopathies, they were all epilepsies in fact, and 1 debuckination deficiency, and tried to run this whole process all the way over for them within the time period we had to see if we could find drugs for them as well. And we actually did it. We found drug candidates for every single disease that we went after in just 12 months. So the question of, can we really do this is yes. And right before I left office, I went back and my final briefing said, yeah, we actually pulled this off. This vision is real. And so we've also been laying the scientific foundation for the Precision Medicine Initiative through the All of Us program. We're recruiting 1 million Americans to donate their phenotypic and genotypic data so that we can start to build what I would call a genetic telescope, which is allowing us to take the human genome and figure out which parts are associated with which diseases and which conditions. And we've done this again and again. So there's actually a second program in the United States, which I've been working very closely, which is the Million Veterans Program, which is doing essentially the same exact thing. And then I'm doing the same thing all over again in Alabama with the Alabama Genomic Health Initiative. So this is my 3rd time setting up a genomic health initiative. And the question of what's next, well, for me personally, again, I remain in the administration and I remain a staunch advocate of science, doing the best I can there. I can't say any more on that. And now I'm serving as the director of the Hugh Call Precision Medicine Institute at the University of Alabama at Birmingham. And I'll give you a very quick summary of what we're doing at this place. And I've really got 3 focus areas. Obviously, rare disease is one of them. And we have a fantastic undiagnosed diseases program where we can take patients today. We have about 180 patients enrolled already, where we are finding answers for them when they had no other way of finding an answer, largely through genomic sequencing. And after that, we're actually doing high throughput drug screening through, to do repurposing. So we can find if there's some other drug out there that works for them. The second big focus area is cancer. My associate director, Eddie Yang, is heading up this, and we're developing algorithms for finding ways to map tumor data into the right medications for them. And we'll take any kind of cancer. We don't focus on any one particular cancer in particular. So if it's brain or head and neck or breast, we'll take it all. We're finding patterns in the genes that drive these cancers. There's certain genes which are repeat offenders and appear again and again and when we sequence these tumors. And the amazing thing is around 71% of our patients have a targetable mutation. So we're running 2 trials right now, and 71% of patients find a matching drug that goes and targets the root cause of their particular cancer. So we'll know in about a year what this means in terms of survival and outcome, but I'm highly optimistic that by addressing the root cause of cancer, we will absolutely improve outcomes. Beyond that, we're actually doing repurposing. So if you have treatment-resistant cancer, we can, and for leukemia in this case, we can actually test any drug out there right now to see if it makes a difference for your particular disease by testing all the drugs that are available against your blood. And the third and final focus area is pharmacogenetics, which I think is broadly applicable to everybody, because we can use your individual genetic profile to determine your response to basically every drug that's out there right now. And the first big area we're going after is actually depression, because depression is sort of a major silent scourge in the United States, and I think across the world. And we think we can short-circuit these therapeutic odysseys where you try one drug after another, after another, from years down to months, and pick sort of the right drug for you right from the start. And the amazing thing is patients are actually showing up. So I'll give you 2 very brief case studies at the end to show what this looks like. One of the patients coming from the undiagnosed clinic had a mutation in a gene called NTRK2. There are 5 patients in the world known to have this condition. It appears to be in a transmembrane domain, or it is in a transmembrane domain, which we think is taking out the function of the gene, but may or may not. And the question is, when you get this information, how do you actually treat a disease like this? And what it involves is taking the algorithm I showed you earlier and instantiating it for this particular patient using the scientists available at an institution like UAB. So I'm mapping that algorithm onto this patient. I'm mapping stages of that algorithm onto individual researchers at UAB. So there's a researcher that can figure out, is this a gain of function? Is it a loss of function? If it's a gain of function, we have a way of finding an inhibitor for it. If it's a loss of function, we have a way of finding an activator. And beyond that, we'll move then on to medicinal chemistry if necessary, and then into an N-of-1 clinical trial for this particular patient. But we already have a way, we already know whether or not it's gain or loss. We have compounds we can give this patient in either case. And then I've been working with an epilepsy patient as well, and this highlights the importance of doing really good variant interpretation. So we found two mutations that look like they could be causing the epilepsy, and the question is, which one was actually doing it? And so to figure out the answer, we actually did computational modeling to figure out whether it was KCN5H, this potassium channel, or RYR3, which is this ranitidine receptor. And by modeling the ranitidine receptor, we immediately saw that the mutation changed a key amino acid that looks like it starts to decouple the protein from the cell membrane. It looked like you could cause this transient epilepsy as a result of that. So we're getting increasingly sophisticated in how it is we actually understand what these mutations mean to figure out how we treat these patients. So I hope what I've convinced you today is that precision medicine is real, and this notion of delivering the right care to the right patient at the right time is absolutely possible, and that it has never been true that this phrase non-actionable was ever something you should ever say to a patient. I think it's absolutely time to eliminate the phrase non-actionable from our medical lexicon, because the event of the non-actionable, it has always been possible and still remains possible to do science. So thank you.